Thanks, Laureen, and I also just want to thank Zephyr and Shanti and all of you for coming out today. No matter how much it rains here, it's still better than the snow that we get in Boston, so we should be grateful. So today I'm going to talk a little bit about, first to give you an introduction into sort of the cohort that we've assembled over there in Boston, and then talk about sort of a potpourri of the different investigative studies that are going on over at the Ragon Institute, some of which I'm involved in, some of which are being done by other investigators there at the Ragon. So the International HIV Controller Cohort is a network of almost 300 clinicians that are primarily based in the US but are international. And we are particularly interested in looking at, in better understanding HIV elite controllers, which are being defined as those individuals with undetectable viral loads for greater than a year off therapy, and also viremic controllers who have detectable viral loads but less than 2,000 copies. This is the distribution of individuals in the US who are participating in the study, and this cohort obviously overlaps and shares many patients with people here at UCSF and at the NIH, and actually, you know, all of the investigators here, we are involved in understanding, trying to better understand the same patients. We also have a significant number of individuals overseas, and this isn't entirely up to date, but that kind of gives you a flavor of sort of the distribution. So in this cohort, there are about 550 elite controllers and close to 1,000 viremic controllers. Those numbers are a little bit out of date, so we're a little bit higher now. The majority of them are men, and the median age is in the 40s, with a duration of diagnosis on average about 10 years. In the elite controllers, obviously, they have median viral loads that are undetectable, less than 50, and in the viremic controller cohorts, the median viral loads are in the 500 range, with pretty good CD4 counts, as you can see there. This is self-reported ethnicity. It's a little hard to see here, but about half the cohort is self-reported as Caucasian, with about 25 to 33% African American, and then a significant number of Hispanics, and then a smattering of other reported ethnicities. Okay, so in terms of the work that's being done at the Ragon Institute, so the immune system is complex. There are many branches of it, and we have people who are studying many different areas of the immune system. So we have Sylvia Legal, who's trying to better understand how antigen processing occurs. So when a cell is infected with HIV, that HIV gets broken down, it gets presented to initiate an immune response. Sylvia is really interested in understanding how that processing occurs. We have Paul DeBacher and Florence and Mary Carrington. They're all involved in studies looking at host genetics. I'll be talking about some of those today. Todd Allen is studying how the virus mutates in response to immune responses by controllers and non-controllers. T-cell immunity, obviously very important. There's been a lot of attention paid to CD8 T-cells. Florencia, David Heckerman, Bruce, and myself, and others actually are very involved in looking at CD8 T-cell responses, and then of course CD4 T-cell responses as well. And then we have groups looking at innate immune responses, as Peter touched upon before, B-cell and antibody responses, dendritic cell function, and also more recently we've begun to look at how HIV affects mucosal tissues, particularly in the gut. Okay, so I think one of the big questions, especially for a group like this, is, well, what do we know about how HIV controllers are control? I mean, what is the mechanism of that? So one simple way of thinking about that, well, you know, is it the virus that's important, or is it the immune response that people make to the virus? And it's not clearly cut and dried, but first let's talk a little bit about the research that we've been doing looking at the virus. So the problem with studying virus in elite controllers is obviously they don't have a lot of virus. So it's very difficult to isolate that virus from elite controllers. So Mark Brockman at our institute has developed a technique where you can amplify small regions of the virus. So you don't get the whole thing, but you get parts of it using PCR, and then you can put that part into a backbone of sort of a standard lab isolate of the virus and generate a full complete virus, and then look to see how fit that virus is. And the details of this aren't important, but basically what you get is a kinetic assessment over 7 days of how quickly that virus is reproducing. So the steeper the slope here, the faster that virus is reproducing and the more fit that virus is. And then you can look at these chimeric viruses from a group of HIV controllers and a group of individuals with chronic progressive disease and get a sense. And what you see is that indeed the virus in elite controllers is less fit than the virus in chronic progressors. So it replicates slower, it reproduces slower. And that's actually true even if you look very early on in infection. So this is a unique group of individuals in which they had samples taken very early on within a few months of acquiring HIV, and then they were followed longitudinally, and some of them had progressive infection, and some of them became controllers. So on the left is the controllers, and what you see is that even early on, if you look in HIV controllers, they seem to have virus that is less fit than people with progressive disease. And from these studies in people who are acutely infected who go on to become controllers, we know that those viruses have a lot of mutations. So some of them have, the virus that establishes the infection, some of them have mutations that cause attenuation or drug resistance. Some of them have what are called escape variants, so mutations that are caused by immune pressure. And many of those patients also express these protective HLA alleles, which we'll talk a little bit more about. And overall, if you looked at that group of 16 or so patients who we had information on early and were able to follow them out and learn later on that they controlled, about 88% of those people had some reason that you could potentially explain why they might control. Either their virus was less fit or they had one of these protective alleles that there has been a lot of discussion about. That's not to imply that it's all the virus, because we know that that's not the case. So just the main point here is just that that virus that is in controllers is less replicative.